All right, so now it's 2 o'clock. So let's start here with what we have to cover. That is going to be also on your first exam on Wednesday. So let me put here in a presentation mode. So before I start on what we're going to cover today, do you have questions or comments? See everybody just starting to get settled there. Yeah, I see with the poll for the exam, most people are going to take it online, but those of you who prefer to go to the class and take it in person, of course, the exam online is going to be available at the same time that the exam class. And for those of you who prefer to go and take it in person, I'll ask Belle to print a few copies of the exam, but you have to bring the blue Scantron. Okay, make sure you bring a blue Scantron and a pencil, of course, an eraser, and an ID. Okay, so we have to check that you are who you are when you hand in the exam. Also, for those of you who are taking it online, you're going to be using a lockdown browser and you need to have your webcam on, okay? Does anyone have questions about the exam? Now, it so happened that I had to change my flight
Sorry,
plans.
someone has a question.
Okay.
If you don't mind holding. He's asking if they're doing it online. It's between 2 and 3:20 still, right?
Yes. Same time as the exam in person.
Yes.
Try to ignore my scratchy dog here in the background.
There's another question.
Yes.
Hi, if I'm doing it down at SAS, do I still have to do online version and honor lock?
No, if you are doing at the SAS, you can either do the paper version or the online version. It's your choice.
Okay, thank you.
You're welcome. But for those of you who have SAS accommodations, I will make sure that you get the extra time at the in the online version as well, so you don't have to worry about that. Any more questions? No, I guess we're good to go. So here, yes, I have uploaded like just now the PowerPoint version for this lecture. And yes, we are still a bit behind, but I don't like to rush through some material that I think it takes a bit more time to digest. So the part that I think is mainly review and you can manage it on your own with the self-studies, those are the parts that I have uploaded. And I hope that by now everybody has seen the self-study sessions and the extra videos and the extra podcasts and everything. Okay, so here, let me put it in presentation mode. And I have to make sure to get my visual devices. They are right here. Very, very sophisticated and technological devices to show planes, but that's going to come a little bit later. Okay, so here, we stopped at this part when we just started to look at protein folding. Okay, so we started to look at protein folding, and we're going to look also at how is it that proteins fold, and once they are folded, how is it that they remain folded? Okay. All right, so first thing you have to understand is that there are several levels for structures of proteins. Okay. So the first thing that you're going to see is the sequence of amino acids that is making up the protein. So that is going to be your primary level. Okay, so primary level is just the sequence of those residues. And again, amino acids and residues, those are interchangeable terms. So one amino acid is one residue when it is into a polypeptide. Okay, so you look at the sequence of those amino acids, that's your primary structure. That's it. Remember that to the left, you have your amino terminus, and then to the right, you have your carboxyl terminus. That's your primary sequence, is one stretched line. And how does it look like? If you're going to look at it, it looks like this. Is a stretched line. That's it. With our groups going from like the top and bottom of it. All right. So here is your primary structure. Let me see here. If we press here, there should be a little video. Yeah. So here it is. Primary structure of your amino acids. And the first one, as you will see, is always methionine, right? So here, this was taken from the genetic code, and joining each of those amino acids, you're going to have your peptide bond. Then you end up with this protein backbone. All right, long backbone, and that's your stretched line. That's it. Once you have your stretched line, then you go to your secondary structure. Now you're going to start to have a little shape to this squiggly line, okay? The most common shapes that you can form are either alpha helix or beta sheet. And we're going to get there. Okay, that was your primary structure. So here, just putting into words what that movie was showing you. That's the linear structure of your amino acid. Again, they are joined by peptide bonds, and that's the direct translation from your messenger RNA. And again, the messenger RNA came from your genetic code or protein coding genes. Okay. Here, the synthesis of that stretch is always starting at the amino end, and the first amino acid is always methionine. Although it's not written here, but you should keep that in mind already. Okay. And okay, so this sequence, this is very important. Okay, so let's make sure that we are all awake and make a note of this. The primary sequence, okay, the primary sequence determines all properties of the protein. So your primary sequence is going to determine how the protein is going to fold and everything that the protein is going to do, okay? Folding of the protein is determined by the primary sequence. Okay, did everybody get that? Yeah, yeah. Let me see hands up who thinks that this is a good test question. Okay, is it going to be a surprise when you see the test question on the test? No, it's not going to be a surprise. And of course, everybody has to call me when I am lecturing. You know, Murphy's Law, right? Okay, so here we go. How is it really that proteins evolved, and how is it that they acquire functions, right? So basically, we know that this primary sequence of the protein, since it is determining everything that the protein is going to do, when the sequence is successful, when you have a successful functioning protein, then of course your system will want to keep the protein for the organism because it can be useful. So when you compare different organisms, evolutionarily speaking, you can look at the sequence of the proteins to determine whether those proteins are related, whether they are conserved, or whether they are modifications of initial protein. Okay, so basically you can see the relationships between the structure of the protein and conserved sequences of those proteins. So comparing two proteins, if they are sequentially similar, you can assume that they are going to have a similar function, okay? Because of course the sequence is going to determine the shape, the folding, and the function of the protein, right? So this is evidence of evolutionarily conserved and determined properties. Okay. So of course, as evolution happens, as we are going to see, as DNA changes, we will see a diversion of certain protein functions. Some proteins are going to work better than others. Some other proteins are going to work worse than the original ones. And the ones that are useful are usually kept for the organism. Okay. So this is a topic, or is a notion that is quite easy and intuitive to understand. If something is useful for the cell will keep it. If something is not useful, for example, if you have a protein that would be vital for, let's say, for example, transcription. So you have a mutation in a protein that is involved in transcription from genetic code to messenger RNA, okay? Let's say something changed with the code of the protein, and now the protein is not working very well for the transcription. Then what's going to happen to the cell? Well, most likely the cell will die, because if you cannot do transcription, then there's no way the cell can replicate, so or make other proteins to maintain its life. So naturally, if something goes wrong with that change in the genetic code, then the cell will naturally disappear. It will die. So this is how you get these evolutionarily distinctions, or you can say that mutations that were not useful got erased or naturally selected. And I really like this picture. I don't remember where I got this cartoon, this caricature from, but it really tells you a lot about folding of proteins. So here you have your protein O contortionist that is really natural at folding. So once it is pressed, it kind of does a little bouncing and folding on its own. It doesn't need anything else to fold in the proper contortionist orientation. Okay. So again, what is important to know here? Proteins that have similar sequences, those are going to have similar structures and most likely similar functions. Okay? And then there's something else here. There are some proteins that have different sequences, but they end up having the same structure, and that would also give them some similar functions. Okay, so now you're saying, yeah, okay, if I know the sequence of the protein, I can predict the structure, but sometimes different sequences are going to give you the same structure. So this final structure business is kind of tricky, because you can have a lot of different sequences for your proteins, but you have a limited number of structures that you can form. Okay, so you don't have infinite structures that are possible, and we're going to see why. Okay. And here, hopefully, this little movie is going to show. And this is showing you what happens to a sequence, to an amino acid sequence, right, your primary structure in aqueous environment. So once it is in aqueous environment, look at this. The sequence, their primary structure folds on its own without any other input or energy input. It will fold on its own to its final lowest energy state structure. Okay, so once you know the sequence, you can predict what is the structure that is the lowest energy for an aqueous environment. And of course, it has to be aqueous environment because life is based in water. Okay. So here, this is just a picture from your book showing the primary structure of your protein. And those little Christmas lights are showing you the R groups, all right? So remember here, you're going to have peptide bonds between each amino acid, and the R groups are going to be either, pointing up or pointing down in this structure. And then once it is in this aqueous environment, what you can predict is going to happen? Well, the residues that are either acidic or basic, they are going to be facing the water because they have certain charge and they like to interact with the water. The residues that are hydrophobic, those are going to be hiding inside the structure of that protein, okay, because they are going to be pushed away from water. So they're going to be hiding inside in the core of the protein, okay? And remember the note that that I put at the end of last lecture? There was a question in class related to what you need to know about those amino acids. Like, do you need to memorize the amino acids? No, you have to have a good idea of the properties of the R groups, okay? You should look at the R group and recognize if it has carboxylic acid or if it has some amino group, and you should be able to tell me whether this amino acid has acidic or basic properties, okay? And you should also tell me whether it's polar or non-polar or hydrophobic, okay? So those basic properties you should know. So you don't have to memorize them, but you should look at the R group and recognize what kind of properties the amino acids have. All right, so folding of that primary structure is going to give you your secondary structure. Now, the question is, what is it keeping the secondary structure in place? What is it holding the secondary structure? So first question, of course, what is holding the primary structure? That's going to be your covalent bond because those are your peptide bonds, right? Now, secondary structure, you're going to have mainly non-covalent interactions, okay? You're going to have electrostatic interactions. So you're going to have van der Waals interactions, and you're going to have some hydrogen bonds, okay? Mainly you're going to have a lot of hydrogen bonds because that's the most important strength, the most important thing that keeps your secondary structure together, okay? So when you look here at your van der Waals interactions, okay, those are kind of weak structures, weak interactions. So you see here the strength of that type of interaction is kind of weak, okay? But that's going to help to keep your secondary structure together, okay? When you look here at what is happening with the atomic cloud, with the electron cloud of those residues, okay, of those side groups, you will see that if they are hydrophobic, they are going to be facing the inside of the protein and in the inside of the structure, and they will tend to interact. Okay, so those van der Waals interactions are showing these mainly interactions of those hydrophobic parts. Secondary bond type of strength that is keeping this together is really a very important one, which is salt bridges. Okay, so salt bridges are only going to happen in the core of the protein. Because remember, when you have ionic interaction, you are not going to have ionic bond in water, in solution, because the ions, the charges are going to be solubilized by water molecules. But inside the core of the protein that you don't have really a lot of water, this is when you start to have those ionic salt bridges, okay? So that is going to be interaction between two differently charged pairs of atoms, okay? So if you look here at the red, that is a lot stronger than your van der Waals interactions, okay? So these are ionic interactions, and it's really happening inside of the protein, outside from the aqueous environment. And then here you're going to get your most important form of interaction, which is your hydrogen bond. Okay, so here you see in blue your, which one is here? The hydrogen bond, is it blue or red? Okay, here is showing this one here. Now, what color is the other one? This ionic interactions. Oh, the ionic interaction is not showing here. I made a mistake there. Okay, so here, this is showing you your hydrogen bonds. Okay, so for the hydrogen bonds, you have to first remember what is it that is causing this type of hydrogen bond. And second thing you have to remember is that this is happening in aqueous environment. No hydrogen bond is going to happen in water. Okay, so this is the most important thing that is holding the secondary structure together, because those bonds, they are between things that are permanent. They have permanent dipoles. Okay, so hydrogen bonding is really what is holding this secondary structure together. All right, so this is really important to remember, which also, of course, sounds like a good another good test question for you. Okay, so here, look what happens with these types of interactions. Okay, so of course, those are all electrostatic interactions, right? They don't have covalent bonds here yet. The secondary structures are not going to have covalent bonds, right? So covalent, you have here what? Ionic interactions. You have here your hydrogen the type of bonding, also showing electrostatic interaction here, but you could have as well this hydrogen bonding here. Okay, here is showing you hydrogen bonding. And then inside the molecule is showing you your van der Waals interactions. All right, so what is going to help to fold your protein? Again, you have to remember your primary structure is going to determine the folding. And now you start to keep in mind, how is this process happening? Once your protein is being made at the ribosome, so it started at amino terminus, the first amino acid comes out, the second one comes out. So the first and the second, they're going to start interacting with each other. And they're going to start turning themselves in a position that is going to be the lowest possible conformation, so that those atoms are not going to clash on each other. Okay, so the first and the second are already going to go and sit in a position that they feel comfortable. And then the third residue comes out of the ribosome, and the third one is going to interact with the second and the first. So the third one is going to be pushed by the other two that are already there. The third one is going to be pushed in a certain orientation to be at the lowest possible energy level for the entire molecule. So this whole idea of interactions here is really, it has really to do with the thermodynamics of the system, and is happening at the same time when that protein is being made. So this is what is meant by co-translationally. Okay, this is what this term means. So the protein folding is starting and is happening at the same time that translation is happening. Okay. Again, at the end of the translation, you're going to end up only with one lowest energy type of folding. Okay. And now get ready, because you're going to get your first question. So get your iClickers out. Just a minute, Bell. Let's read this first. So you get ready to answer when we press, the iClicker thing, all right? I just gave you the answer. When does folding of a protein begin? So options. Begins co-translationally as it emerges from the ribosome. Ta-da! Okay. It begins post-translationally after it emerges from the ribosome. It begins after it emerges from RNA polymerase. Who is that guy? All right, so let's start the time, and let's everybody get your couple of points here. Actually, this is the first question, so you end up with 3 points.
Okay, 30 seconds are up.
So what is your answer?
Most people answered A.
Yeah, that's your right answer. A is the right answer. I just gave you the answer. And of course, it's really easy now because I just mentioned it, right? All right, so here is one of the slides that you absolutely have to remember. And I just mentioned hydrogen bonds. They are the ones that are really important, holding your secondary structures together. Then you're going to see, well, how is that happening? So let's see here. When you look at the alpha helix, okay, alpha helix, and you're going to look at residues that are not too far away from each other. At the backbone of this alpha helix, you're going to find that you're going to have hydrogen bonds that go up and down like that. Okay, so hydrogen bonds going up and down like this, those are going to hold your secondary structures together. Okay, alpha helix is holding like that. And now when you look here at a beta sheet, you'll see that it is depicted like this, like if it is amino terminus here, carboxyl terminus there. So the arrow is going to point here to the carboxyl side. And then this other arrow is showing that most likely this thing turned around here and is going that way. And then this thing goes back this way. All right. But you have several arrangements of beta sheets that are possible. When you look at the beta sheets, you're going to see that between these two primary structures here, that's where you're going to find those hydrogen bonds, see? And they can be also of a stronger type of interaction or weaker. Some hydrogen bonds are going to be like really straight, and some other hydrogen bonds can be a bit turned to the side, okay? But they're going to be between those two primary structures that are closest to each other like that, okay? And now we're going to start seeing how is it that you can predict the shape of those secondary structures. Okay. Remember that all amino acids are with the L conformation, right? They all level rotatory. Okay. So when you have your primary structure that is starting to form that shape, they are all going, if you have alpha helix, they are going to be in this right-hand conformation. Okay, so you're going to have right-handed helix that is going to come out of there. And remember, you're going to have those hydrogen bonds, right? They're going to keep it together. And usually those hydrogen bonds, because of the size of your peptide bonds, the hydrogen bonds will have to be a certain number of residues away from each other. So usually for the alpha helix, you get the hydrogen bonds that show up every fourth residue. So that's what is meant by N+4. Okay, and is the number of residue or residue number. And then N+4, you get your hydrogen bond over there. Okay, so you see depicted over there. Okay, so the peptide bonds are going to be arranged inside of the helix, and then as we're going to see, the side groups, the R groups, are going to be pointing to the outside of that helix. And we are going to look here at this plot that is called the Ramachandran plot. Okay. And we're going to see, what this means. But basically, when you have alpha helix, you have a certain type of conformation of your peptide bonds that allow it to fall at certain angles to allow that torsion to fall into this type of helical shape. Okay, so here's your ribbon description of your alpha helix. Okay, so your peptide bond is going to be to the inside, following the ribbon, and then your side groups or your R groups are going to be pointing to the outside of that ribbon. And again, this is depicted green, showing you that this is the area of alpha helix distribution for angles. We're going to get there soon. All right. Now, another thing that is important here, when you have hydrophilic residues, those are going to group in one side of that ribbon. When you have hydrophobic residues, those are going to get together on another side of that ribbon, because those are going to further fold when you get to the tertiary structure, and they would like to interact with each other. Okay, and same thing with the hydrophilic residues. They are going to interact with each other and they're going to push the hydrophobic residues like to the other side, away from your hydrophilic side. Oh, another question. Where do you expect to find the hydrophobic region of a cytosolic globular protein? So now what you should do is draw a blob of protein. Okay, so let's do that before you start the thing. Here is your blob of protein folded properly into this contortionist model. And here you're going to have certain residues. And then on the outside, you're going to have other residues. So this is all in water. So water all around. Now let's start the question. The eye clicker, and now you should answer. Is it going to be facing the cytosol? Is it going to be away from the cytosol? Insulated away from cytosol? Or is going to be anywhere because this thing is a mess in the first place?
Sorry, what?
Time's up?
Yeah,
Huh?
time's up.
Oh, okay.
Time's
So
up.
your answer is?
Most
Hey,
B.
everyone. B, yes, it's going to be isolated away from the cytosol. And yes, I am quite generous. I just give you the answer so you get all the points. And so they know if you showed up for class and you are awake, you might as well get your points, right? So how you visualize your secondary structure, okay? That's when we're starting to get here to this Ramachandran plot. And we're going to get into more details on how this was elaborated, okay? So you have certain angles. There are phi and psi, and those are angles that are related to your alpha carbon, okay? So here, let's see, here is your alpha carbon, okay, right there. So you have a phi angle that goes this way, close to your carboxyl group, and you have your psi angle that goes towards the amino site. Okay, so here is your this is peptide bond. So you have here, okay, alpha carbon, this angle, that angle. Now remember, the alpha carbon here is showing you this R group, is not showing you the little hydrogen, right? So the alpha carbon is connected to carboxyl, amino, hydrogen, and the R group. Now, these two bonds here, those are the ones that are single bond, not your peptide bond, okay? These are the ones that are single bond, and they are free to rotate, okay? So peptide bonds, not peptide bonds, the phi and psi bonds are free to rotate. When this bond rotates, the R groups, okay, so this is the peptide bond, this is the peptide bond right there, okay? The R groups are going to be placed away from each other in a space that they can feel comfortable. They don't like to be cramped together. Okay. So the angle of that bond is what is showing over here. If your bond, let's say, if your psi bond is in this case between these two different angles here, then most likely you're going to have alpha helix. If the bond is like phi bond is between those two angles, you might as well have a beta sheet type of configuration. Don't panic yet. This is going to get clear in a minute. Okay, but there are certain allowed angles that are going to make your residues fall either in an alpha helix conformation or in a beta sheet type of conformation. You get better pictures of that. Okay, so let me see here. Okay. Besides the alpha helix and beta sheet, there is also another area that allows for freedom of movement, that is capable of avoiding clashes of electrons, and that is called a loop type of domain. So you have alpha helix, beta sheets, and loop domains. Let me get to the slides that are going to make this idea to be a lot clearer. Second. So besides those alpha helical and beta sheets type of structure, that those are very common in globular proteins, the ones that are floating in the cytoplasm, you have this other type of configuration that is basically two-dimensional. It's basically a fibrous type of conformation, okay? And it's a more simpler type of arrangement, but it also has the same cohesive properties that we have seen before. Basically, what keeps this together is, again, electrostatic interaction, okay? So this type of proteins, they're called fibrous proteins. They're very important for structural maintenance of cells and tissues. And basically, as I mentioned, they have only a 2D type of structure. You don't see a blob out of those proteins. The example of these proteins are really like collagen or keratin or something that is really, really fibrous and more used for maintenance of structure. So when you look at this type of collagen structure, for example, you see that you have helices, right? You have alpha helices that interact with each other. And in this case, you have 3 of them. Here is showing 2 of them, but usually have 3 of them that coil around each other, and they interact in a way that hydrophobic residues are going to interact with each other, and then hydrophilic residues are going to be facing the aqueous part, okay? And this interaction is what really keeps this structure in place, okay? So we're starting to look at this idea of secondary structure, right? So again, primary structure is just a sequence. Secondary structure is that sequence is starting to fold and form either alpha helixes or beta sheets, okay? And the type of structure that you find in fibrous proteins is also a secondary structure. It doesn't have the complexity that you see with tertiary structure. So now we'll get to the tertiary structure. And the type of interactions that happen in tertiary structure is basically the same as the type that happens in quaternary structures, okay? So here we are going to see that you have further folding and interaction of these individual alpha helixes and beta sheets. So these individual domains are going to fold and start interacting with each other. So now you're going to see alpha helix interacting with beta sheets, or 2 alpha helix interacting with each other. So this is going to give you a more organized tertiary type of structure. And now what is important for keeping this together, again, those electrostatic interactions, but also you're going to have the presence of some covalent bonds that are possible because of sulfur bridges, okay? So in tertiary structures, you can also see some covalent bonds between 2 domains. So when you get these tertiary structures, then you can see you have here alpha helix here, another one there, and beta sheet here and there. So the folding of those domains, they are going to, they say, okay, so actually motifs, the folding of these motifs, they're going to start forming the domains of a protein, okay? So, for example, you can see certain structures that are common and recognizable in several globular proteins. For example, you can have alpha helix in the middle of 2 beta sheets. So this is going to have like beta, alpha, beta type of motif. Okay. And then here, when you put motifs together, then you start forming a more elaborate structure, which is a domain. And domains are very common for globular proteins. Okay. So here, this is a definition of a domain. Domains are really independent folding units. All right, so you have a lot of blobs that are predicted to fold in certain forms and they have a certain function. So this is called a domain. Motif is something that is predictable, but it doesn't really have a function. Okay, and it's smaller than the domain. Again, here, let me emphasize here because a lot of times this gets to be confusing. Okay. Domains that are going to interact with each other, they're going to form here a functional unit, okay? Domains form functional units. All right, so here, when you look at globular protein, they have several domains. Okay, so this is one type of domain, that's another type of domain, and those domains are very common among many proteins. Okay, so you put several domains together, and then they form this functional unit. And this one is showing you, what is this molecule carrying ATP? I don't remember which molecule it is, but yeah, it's basically, it's carrying ATP here, so it might be phosphatase or something like that. But it is showing you that it has some function. So here is just a little summary of the different structural levels, all right? So you start here at your sequence of amino acids, and then you're going to have here your secondary structure, okay? Remember, what holds secondary structures together, those are your hydrogen bonds, all right? When your structures start to fold on each other, you're going to form your tertiary structures, and tertiary structures, they can have disulfide bridges, and that is your covalent type of bond, okay? The only way that you're going to get disulfide bridges is if you have two residues here of cysteine, okay? So that is important to know. So cysteines can form disulfide bridges that help to hold the tertiary structure together, not secondary, okay? Tertiary. Then you put several tertiary structures together and you end up with your domains, and that's your functional unit. And another question is coming up. Okay. Which is true of amino acids classifications? Cysteine contains a sulfide hydroxyl group. That's the only one in this category. Amino acids cannot be classified in more than one subdivision. All amino acids show L chirality. Ooh, I didn't mention one thing about one amino acid. So let's cross C as a wrong answer. Okay, that's not the right answer. Okay. Hydrophobic amino acids are commonly found on surface of enzymes. All right, you should get the right answer out of this 4, knowing that C is not your right answer. Okay, let's start the timer. Time's up.
Okay. Yeah.
Okay, so what's your correct answer here?
Most people said A.
Yes, A is the correct answer. Can you tell me which one is the amino acid that does not have chirality?
Someone said glycine.
Glycine, why? Can anybody tell me why? Is this CO2 here? I don't remember. So this is H up here. So this should be glycine, okay? what is going to happen here, COH, sorry, what am I doing here? So you're going to be your carboxylic side, COH. What is happening here, when you look at glycine, your alpha carbon is not an alpha carbon because it's not connected to, it's not a chiral carbon because it's not connected to 4 different groups. So if your carbon is connected to 2 groups that are the same, so it loses chirality, it loses the mirror image. So glycine is the only amino acid that does not have that mirror image. Okay, so it's not L and it's not D, so it does not have chirality. So here is a slide showing what happens with those disulfide bridges. All right, so cysteine is the residue that can allow for this type of covalent bond interaction. All right, so those are your disulfide bridges, and those are important for keeping your tertiary structure as well as quaternary structures together. Okay, so here is a beautiful picture of hemoglobin. Okay, it's showing over there. And you see here, hemoglobin is showing quaternary structure because you have several tertiary structures together. Again, what is very important for a tertiary structure is one long polypeptide chain. Okay, to make a tertiary structure, you need only one chain. To make a quaternary structure, you need 2 chains that are going to be interacting with each other. Okay? Tertiary, only one long chain. Quaternary, more than one chain that will interact with each other. Make sure that you know that distinction. That's the only really difference between tertiary and quaternary. And here is showing you a protein folded that is interacting with a DNA molecule, okay, in this type of shape, this type of conformation and domain. Okay, so let me see here other quaternary structures. Well, we see proteins that can form complexes with other types of molecules like protein and RNA, like we see in ribosomes, right? Also spliceosomes. You get proteins that are interacting with lipids, like for example in membrane. Okay? And remember the type of interactions that they have. Okay, so let me see here on domain structure. Okay. It's just a little summary of what I had mentioned. Specific function. So a domain again will give you some type of function. In this case, you can have your binding domain. Okay, of this type of protein is like a zinc finger that interacts with DNA. So you can distinguish what kind of domain will do what. And here's again showing you a type of protein that is carrying a molecule of ATP. And you can, if you know what kind of domain you're looking for, basically you can distinguish which domain is helping to fold this huge complex. So here, let's see how is it that proteins have evolved naturally, okay? So how is it that you're going to find usefulness for a certain protein? So basically remember that you're going to have to find a final conformation that is the lowest possible energy conformation. Okay, it's the lowest possible energy that the structure can get into. Okay. Another thing for a protein to be kept in a system is that it has to have a function. If you just make a protein and it has no function, that is waste of energy. So cells don't like to do that. Okay? And that function of the protein has to be predictable. That protein has to do exactly the same thing that it was intended to do at first. So they have to be at the lowest energy. They have to have the function. The function has to be reliable, has to do the same thing all the time. Okay. The thing that the protein is doing cannot kill you, all right? So that thing has to be useful for the metabolism of the organism, okay? Usually when you have mutations, the end product doesn't really achieve all of these types of requirements, okay? So usually when you have some mutation in the gene, the end product is a messed up protein that is totally useless, and usually that messes up the cell and the cell will die. So that will contribute to the natural selection or natural death or something that is not useful. Okay. So now we're going to get to this part of understanding the atomic structures of proteins. One second, I think I put the order of this in a strange arrangement. Let me just confirm one thing here before we go. Keep the annotations. Okay, let me just confirm one thing here. Oh, I see what I did. Okay, yeah, I opened, this is what happens when you're running like crazy. I did open the wrong file here. I was missing something. Okay, hang in there. Because this whole idea of the Ramachandran plot that you absolutely have to know, it was missing in there. So in my head, things are not quite making sense. Just hold on a minute. We'll fix this in a second, and we just have enough time to fix it. So bio 5 and 6. This one here is that. I apologize for the mess here. Because I don't want to get into protein function before covering, of course, the basics. Oh my goodness. Yeah, I would give you guys some 5 minutes break, but I know I'm gonna find this very quickly, so I don't wanna really spend 5 minutes. Just, well, if you wanna stand up, stretch your legs while I'm looking for this thing that is really important that I have to find. You can stretch up your legs for a little while. Would help a lot if this thing would be a bit faster. Yes, everybody's quiet. Nobody wants to stand up and stretch. Okay. That's pretty low on the background. Okay, I got it. All right, so I have reorganized my thoughts here, and this is the part that you're really missing. There should be before we started talking about, primary and secondary structures. Yeah, I do apologize for my my lapse here. So this is paused. My screen pause is showing. Okay, so let me share this again. And let me make sure that we have this record. All right, so let's put ourselves back together here to where we stopped on this thought here. Last time that we had talked about.
Com Amazon Music Unlimited, é possível pedir para a Alexa tocar as músicas
Quiet, Alexa.
instantaneamente.
Okay. So we had talked about the properties of amino acids before, right? So let's just recap here what we had before, okay? And remember that before we started talking here about the primary structures, secondary structures, and so forth, we had gone over the individual amino acids, and we had gone over the carboxylic ones that give you acidic properties, and the ones that give you basic properties, and the polar and non-polar. And I remember here we stopped at this slide, this lecture, this part, talking about cysteine, right? So everybody remembers this guy here? Everybody remember cysteine? Let me just do a wake up check. Are you seeing the same thing that I have here with the cysteine? What is showing there? I don't see
No,
it.
no, I think it's a different PowerPoint, Dr. Waziri.
Oh my goodness. Okay, let me make sure.
We're still seeing like the atomic structure of proteins. That
Oh.
slide.
Close that one. 1 second. So now I stop sharing. So let's go again here. Do this sharing here. All right, now you should see.
Yeah, now it's the right one.
All right, so now you see here the structure of this sulfur containing amino acids, right? So let's just go for the wake up check again. So everybody remembers properties of amino acids that are acidic. Who remembers the acidic ones? Acidic amino acids, those are gonna have negative charges. Basic amino acids are gonna have positive charges. Then you have the non-polar and the polar, right? Non-polar amino acids or hydrophobic amino acids, they have carbon backbone, right? And I did not cover this slide here before, right? So this here are two amino acids that are quite special. They both have sulfur in them, right? So you absolutely have to know them, right? One of them is methionine, and methionine is the first amino acid in protein synthesis. It's always going to be methionine, okay? But look where methionine is. You have the sulfur right here that is between two carbons, and this sulfur here is quite non-reactive. It makes this molecule polar, okay? It makes it polar, but is non-reactive. So this thing is useless for making disulfide bonds. So what do you have to know about cysteine? Sulfur is stuck in the middle of two carbons, is non-reactive, and is the first amino acid in protein synthesis. Okay, everybody got that? Yeah, not, all right. This other one here that is very interesting one, that is cysteine, right? So cysteine, look here. Your sulfur is at the end and is connected to a hydrogen. So this is the one that can form those disulfide bridges. If you get two cysteines close together, they have a good chance of interacting and sharing those electrons, and here, getting rid of the hydrogen here, and you're going to have your disulfide bridge, okay? So two cysteines can form disulfide bridges. And that is important for maintenance of tertiary structures as well as quaternary structures. Okay, everyone is clear with that? Because I always like to ask questions about cysteine and what is the use of that. Okay, all right. Remember we mentioned before that your peptide bonds, those are dehydration reactions. That means you get a water molecule out of it to make the bond, right? And dehydration is also called condensation reaction, right? So everybody should be very familiar with your peptide bond. Here, that's the part that was really missing for you to totally understand what's going on with the electron clouds and Ramachandran's story, okay? So let's tell the story a little better so nobody is confused about it. So here is a residue that is going to get together with another amino acid. They're going to undergo dehydration reaction and they're going to make the peptide bond, okay? Let's see what's going on here. First of all, where is this happening? At the ribosome, okay? Ribosomal RNA is responsible for the catalysis of that condensation reaction. Important, all right. So here we go. When you make a peptide bond, that peptide bond is going to have resonance characteristic, okay? So what does that mean? So let's see here. Carboxylic acid is going to be close here to your amino group, okay? So here is your peptide bond right there between your carboxylic acid and your amino group. So make sure that you know what a peptide bond looks like. It's right there, okay? It's this right is like right there. This is your peptide bond right here. And this is your peptide bond right there. But look at this. At this side here, it shows like a double bond. And on the other side here is showing like a single bond. So what's going on here? Remember, your peptide bond is between your carboxylic acid and your amino side, okay? And then you have here alpha carbons around that. So let's see what is happening here. This is important to know. This electron cloud around oxygen is not showing, but you have two pairs of electrons right there, right? This electronegativity of oxygen, sometimes it likes to hold on to the electrons of one of those double bonds, and then it holds on to it for long enough time that it gets a negative charge. Okay. When this thing holds on to the electron, what happens is that the electron pair that is right here at the nitrogen, the electron pair will be pulled to this peptide bond. So that electron pair is going to jump here and be shared between the carbon and the nitrogen, okay? Because carbon has to have 4 bonds. It's in a molecule, it has to have 4 bonds. It's tetravalent, right? So what happens here, when oxygen pulls the pair of electrons, the nitrogen donates the pair of electrons to keep carbon at 4 bonds. And now this peptide bond gets a temporary double bond character. Everybody got that? Temporary double bond character? Let me see nods. Nods? All right, great. I see one great excited nod over there. All right, cool. So temporary double bond character. That is the property of resonance, okay? That gives a strange type of characteristic to this bond that makes it non-flexible. Peptide bonds cannot rotate like single bonds in the carbon. So a peptide bond is going to be always flat. The molecules attached to peptide bonds, they cannot turn around because double bond is not flexible. They do not rotate, okay? So what's happening here? Now you're going to see that when you have your different amino acids, right, together by peptide bond, you're going to look at the R groups of those amino acids. And you're going to find that most of them are kept away from each other. Because look what happens. If you have the R group that is flipped to the other side, in this peptide bond, you know, it's rigid, it's not going to rotate, then the R groups are going to hit each other. They're going to clash up there. You cannot have electrons occupying the same space. So cis conformation is not good for those amino acids that have these R groups, okay? The only amino acid that doesn't have this issue is glycine, because glycine doesn't really have a side group. It has a hydrogen. So hydrogen is small enough to be rotating around, to be either one side cis or trans, okay? But most amino acids, those are going to be in a trans conformation, all right? So what is a cis conformation? This is a class exercise. Side groups are your arms. Cis conformation is both groups up, right? Everybody go cis conformation. Cis. I don't see arms. Do you see me in the video, or am I talking to myself here? Yes? Cis. Trans. All right. Cis. This is a class exercise. Everybody, cis. Trans. Cis. Trans. All right. This is a quick review of organic chemistry. Okay. So now the stuff gets interesting. Because here we go. Here is the business with the psi and the phi that I was talking about Ramachandran, and you're talking what kind of Greek or Indian word am I saying, right? Here, peptide bonds are not going to rotate. That means the peptide bond is going to hold all those atoms that are involved in the bond. They're going to hold them in one plane, okay? So look at this peptide. The peptide bond here is going to hold nitrogen, hydrogen, carbon, oxygen, another carbon, at least 4 atoms that are going to behave to determine that plane. But really, the plane of a peptide bond really holds 6 atoms in a plane. Okay, if you count them, 1, 2, 3, 4, 5, 6. But you really have 4 core atoms that are being held in a plane. Okay, so here in this peptide bond, you have one plane. Then you have an alpha carbon, and then another peptide bond here, you have another plane. So this is where I get my visual aid to show you. This is a plane. This is a paper bag. Everybody can see the paper bag? I cannot see myself in there. Can you see me at all over there?
Yeah, we can see it for the most part.
Oh, okay. So here is a demonstration of what happens to these planes, okay? Here's one plane. So in the middle here, you have your peptide bond. These are atoms held together in a plane. And now here, you have another plane. And my body is the alpha carbon. So my head here is the C for the alpha carbon, okay? The bonds of my arms that are getting together with these 2 planes, okay, my arms can rotate, but the planes cannot rotate, okay? You would, see if I can throw the planes better here. Here are the planes, right? So the planes are here. The planes can go this way and that way. So to my right side here, I have my psi bond. That's my psi bond going back and forth. It can go like 0, 180 degrees. Well, my arm is not going to turn around. Okay, 0, 180 degrees of the plane. And then my psi arm here on the left side, okay, my phi on the left side, it can also rotate that plane, okay? So 0, 180, and 0, okay? For these 2 planes to be in better, lower, or lowest conformational stage, they have to be at a trans configuration, okay? Otherwise, the side groups are going to clash on the top or on the bottom, okay? So the planes are going to be away from each other, and only my arms, my humerus, is going to be allowed to rotate. So phi and psi rotate, but the planes do not rotate. Right? So I hope that was a better visual. What is the sharing stop? Okay, this thing is acting weird. All right. So let's see what is happening here with these configurations. Okay. So you see here that certain angles of phi and psi, they are not possible because when these bonds go in cis configuration, the electron clouds would hit each other, and that is not possible because 2 atoms cannot occupy the same space. okay? And this is very important for calculation of hindrance among those groups, among those atoms. Okay. So this is actually where you get this Ramachandran principle and folding of secondary structures, because the ability of those electron clouds to go away from each other, it's so strong that it's going to determine how that protein is going to fold when the amino acids are being put together at the ribosome, okay? The phi and psi angles, the only ones that can rotate, those are called dihedral angles, okay? The dihedral angles, they really determine how that protein is going to fold. Right. So here, when you see your peptide bond, you're going to see that those are going to be your flat planes, right? And the ones in the middle, those are going to be your alpha, your phi and your psi. Okay. So phi and psi can rotate. So here is showing the rotation that can go all the way around from 0 to 180 degrees. And that's easy to visualize. Okay, so I put several movies here that's going to help you to visualize what's going on here. Okay, so your phi angle, here's a peptide bond, the partial double bond character, okay? Phi can rotate. So how is it that it can rotate? So phi, look at this, goes from 0 here, or 180, right? Let me see this movie again. You don't need to know like which side is which. I just want you to see here. Okay? Your phi angle going from 0 to 180, all the way up there. Going from 0 to 180. Okay? And then if it goes down the other way, or if it goes down this way, it can go on the negative side. So negative 0, negative 180. You could say 0 to 180 back to 360, but they like to put either positive 180 or negative 180. So that's just, minimal. The main idea here is that you absolutely have to know about those planes that are non-rotating around your partial double bond for your peptide bond, okay? And you have to know which ones are the bonds that are allowed to rotate. And then here is the idea of these two, paper bags rotating around the arms, okay? And here is your alpha carbon there. So these planes are rotating around these angles. So here is showing you the electron cloud around the atoms involved in these peptide bonds, okay? So you see it's very, very bulky. So if you see a ball and stick, it looks small and it looks like you have a lot of possible angles. But in reality, these are very, very bulky molecules. So when you look at the possible angles that these things can rotate, you really don't have a lot of choices, because look, if they go at a certain angle, that electron clouds would be occupying the same space, that's impossible, okay? Because electron clouds are not going to occupy the same space unless they share the electrons, and that is called a covalent bond. And you don't have covalent bonds when these things rotate. The covalent bonds are already determined. So those electron clouds cannot go through spaces that they're going to be fighting for the same space. Two things are not going to occupy the same space. When it seems like they're going to occupy the same space, that's an impossible angle. It's this when you're going to have a clash. Is that concept clear? Okay, how many people got the concept? Two things cannot occupy the same space. Let's raise a hand. All right, that's easy. Two things cannot occupy the same space. So let's see what happens here. Okay, so now you get to this plot. So this is telling you at the x-axis, you're going to plot possible angles for phi. At the y-axis, you're going to plot possible angles for psi, right? And you got to remember that those are the ones that can rotate, and they're not the partial double bond character. They are single bonds. So here you see that for phi, you can have an area here in your x-axis that can go from like negative, I don't know, 45 to around negative 100, like here in your x-axis. And then for psi, you have some area that you have a lot of probability that goes from more or less the same type of, the same number of angles. And here you see that this high probability, when those two residues fall into those angles, you're going to have formation of alpha helices. Now, there are other high probability sessions here, and when those angles are falling into those coordinates, most likely you're going to end up with a beta sheet. And then a third area of this graph is where you're going to have your turns, okay, your beta turns. Okay, so now things should be a lot more clear, because look what happens here when you have your phi angle clashing. When it clashes, that's the area of low or no probability. If it clashes, it's not going to hang around there because it's impossible to be in those angles. Okay, so white areas means those are impossible angles. Okay, so here, let's see here, see the other one, your psi on your y-axis. You have two areas that are high probability, and the rest of the angles, those white spots, those are when you have the clashes. So that's like no probability. If it clashes, forget it, it's not going to go there, right? So if you have from positive 10 to negative 55, like here, that is alpha helix. And when you have 90 to 180, that's your beta sheet. So everybody should know the coordinates, like how to calculate if it is beta sheet or alpha helix. So that should be simple enough for that. Okay. Okay, so we have seen that. All right. So that is your Ramachandran principle to determine whether you're going to have alpha helix or beta sheet. And now you should be thinking, what is it that I can possibly ask in terms of these notions of these topics, right? So here, another example showing that, look up here, You see the angle, if it is at a side that is impossible, there's going to be on the white side here. Now, if it is on the blue side right there, or the green, that's when you're going to have either your alpha helix that is here in green or your beta sheet. Okay. So here is the idea that you have two major areas in which you can have either one or the other secondary structure. Okay, is everybody clear with the Ramachandran plot? You should be very comfortable with that. So before you go here, you do have another question that I just had mentioned. How many bonded atoms are required to constitute a dihedral angle, right? Such as the phi or psi. I had mentioned that. Let's see how many was going to get it right. Is time up?
10 more seconds. Okay, time's up.
Okay, what's your answer?
Most common answer was C.
Yes, C. That is the right answer. Now, if I ask you how many atoms are involved in a plane of a peptide bond, then your answer is 6. Okay, so we're going to stop here, and whatever I mentioned until here, that's going to be on your test. So I will hang around for some more time.
The poll that you sent out an announcement, was that to determine, is it the majority that gets the answer, it's how to do it, or?
No, it's just individual. Like if you want to do it in person, you can do it in person. If you want to do it on.
This taking its time, just a second. Share, okay. So this one that you're asking,
I think, yeah, it could be that one or like one of the a few ahead.
okay, this here, can you see my mouse here?
Yes.
Can you see the cursor? Okay, so when you look here at the alpha carbon, right? You see here to the left, you're going to have here your amino side, right? To the right, you're going to have your carboxyl side. Now, if you go further, carboxyl amino, that's your peptide bond. So this one here has double bond, partial double bond character, right? So if this one has partial double bond, this one is not going to rotate. So this is going to be stuck in a plane. How you get the partial double bond with this electron cloud that goes up to this side, and then this electron cloud that is in nitrogen makes this double bond temporarily, okay? So if these two atoms are stuck in a plane, then this angle cannot rotate, but this other angle here is not stuck in a double bond character. It's only single bond character. So this angle here, this bond can rotate around. So when this angle rotates, when this bond here rotates, the whole plane rotates with it. So this is why I had the paper bag showing the plane. Okay? But now the plane here, let's say my elbow is the carbon, okay? The carbon, and the plane here, my humerus is psi, okay, is a psi angle. So my humerus rotate, and when it rotates, the rest of the plane rotates, okay? But the peptide bond that is holding this whole bag in a plane, that bond itself does not rotate. The bag is not going to twist, okay? But it's just going to rotate using the arm that is psi, that's the one that can rotate. Okay?
Yes. Yeah, so to
Okay.
make sure the peptide bonds like don't rotate because of like the temporary
Yeah.
like double
Partial double bond, partial double bond character. Yeah.
Oh, thank you. And yeah, so the test is on Wednesday, same time as a class.
Yes.
Right. Okay. About how many questions?
50.
50. Oh, right, right. Okay.
Mhm.
And then the PowerPoint slide will be updated after, correct?
Yeah, I'm going to make sure that I have everything in place here and I'll update that.
Okay. Thank you.
You're welcome.
Dr. Waziri,
Yeah.
first of all, if I can't make like your normal office hours, is there anyone who's had like made an appointment with you that
Oh
I
yeah,
can
yeah.
join?
Just yeah, drop me an email. What time can you make office hours?
So would it be the same day as your normal office hours? Like would it be today or
Yeah, we can make other Zoom call for later on.
okay,
Uh,
okay. It would be any time from 5.
Okay, so now it's 3:30. So say 3:30 to 4:30. How about 5:30?
5:30, and it couldn't be in the week coming, right? It would have to be during the day of your office hours.
Uh, this time I'll say yes, because next
Okay.
week is gonna be totally insane for
Busy.
me.
Okay.
Yeah, actually, yeah, I had to change my flight plans again, and I will be like at the airport at the time that you guys are taking the test. So I will try really hard just to be on my, my phone, at least in the Zoom. But
Okay.
yeah, I'll be waiting to go through security and everything at that time. That's gonna be a pain. So
Okay.
yeah,
So 5:30.
today 5:30. I'll do today 5:30.
Okay. Okay. Thank you.
You're welcome.
Was that it? No
That
problem.
was it.
Okay, I think that was all the questions. Oh, I had a question, I think. What do we have? Is there something going on Monday?
Sorry.
Is there something going on Monday?
What do you mean?
I don't know. I thought I saw like in the modules that there were like slides that they had to go through or something that they had to go through. And I think last class you mentioned it was on Monday, and I was a little confused because I know we don't have class on Monday.
No, yeah, I probably got confused with my other class. Because yeah, it's a Wednesday and
Okay.
Friday, and yeah, and I
Yeah,
give like one lecture after another, and I know sometimes I like, am I on AMP, am I on cell bio? So it's like I mentioned Monday, but no, test is on Wednesday.
I got you. And for the exam, do you want me to like I don't need to like start a Zoom, right? Like if because I'm going to be proctoring it in person. You said you're going to be at the airport. I don't know if you wanted me to start
Yeah,
a Zoom.
there should be, one second, let me see. Let me stop the share here. And let me pause the recording.